Good evening. My name is Pani de Cossard. I'm presenting today for the channel Quantum Origins. The title of my talk today is The Origin of Species. But to be more precise, we'll be talking about both the origin and evolution of species. The origin of species is the concept we have retained ever since Darwin, but the one that will be more fitting for our talk today is the origin of lineages. We'll be talking more about lineages than species. And we will understand why it should be so during the course of this presentation. And we'll be talking specifically and only about the origin and evolution of species in the domain of life, Eukaryota. And there are three domains of life according to Carl Woese. That's his theory. Ever since his works in the 1970s, he proposed, he divided the tree of life into three domains: bacteria, archaea, and eukaryota. And he's actually the discoverer of the domain of life, archaea. But we'll be focusing our talk only on the origin and evolution of species in the domain eukaryota. Carl Woese was an American microbiologist. The problem we have tasked ourselves to solve today is the following. To find the manner in which life has generated so many species, whether we found them in the African savanna, in the Amazon rainforest, in the poles, the Arctic or the Antarctica, the many species of plants and animals we find in the ocean, plants such as seagrass, phytoplankton, the algae, and different animals in the ocean. And the many species of beetles, we know there are many of them, and about 350,000 known species of beetles. According to some scientists, beetles make about a quarter of all the species of animals that we have. And one biologist of England, a British microbiologist, J.B.S. Haldane, did say that God must be very fond of beetles when he was asked the question what the study of life could say about the Creator, about God, and his quote was, "God must have an inordinate fond of beetles." Up until the publication of my book, Ways of Nature, we had two main contenders about the origin of species. First, the Bible in Genesis 1, where it said that God created living organisms after their kind. This was understood to say that the species were created individually and separately by God. It was a supernatural creation. And in Genesis 1, chapter 1, verses 4, it said, God said, "Let the earth bring forth the living creature after his kind, cattle and creeping thing, and beasts of the earth after his kind," and it was so. And this was until the publication by Darwin of his book on the origin of species, where he presented his theory of natural selection, according to which he says that the species that we see today, they originated from one or few common ancestors in the distant past, from several million years ago, and those would have diverged and multiplied and diverged and pruned by natural selection to make the species that we have today. So he took the opposite view of the church, from which the church says, from the Bible, species were created, each of them individually by God, and himself he said that there was one or a few common ancestors that diverge over time and create the species that we have today. So the basic formulation of his theory is that initially we have one or few common ancestors, and there is Subpopulation, overproduction of offspring, and those offsprings would be different one from each other because there is random variation, and those variation would be transmitted. There is an element of irritability, and that this tapestry of variations would be born by natural selection, and we'll end up with the species that we have today. And here this is the only figure, only diagram that we presented in this book on the origin of species. At the top we have the species that we know today. They would form divergence from species at the bottom. All the way to the bottom we have one of few common ancestors that would over the million of years, several million of years, diverge and up until today, and all that is born by natural selection. But we would be remiss if we did not include Alfred Russel Wallace in the credit for this view of life, because Alfred Russel Wallace was a naturalist as well who was interested in this topic, and while he was exploring the Malay Archipelago, which is modern day Indonesia, he wrote an essay with similar ideas and sent it to Charles Lyell, a geologist of Victorian England, for publication. It was this essay actually that prompted Darwin to rush and publish his ideas in his book Natural Selection, in his book on the origin of species. Darwin's conclusion was that, contrarily to the view of the church, the species were not independently created, but instead they descended with modification from a single or a common ancestor or a few, and that was born by natural selection. One of his quotes is that I view all beings not as special creations, but as the lineal descendants of some few beings that live in the distant past. We have come now to understand that the species did not originate according to the theory of natural selection of Darwin. There are many challenges to his theory, which we can read about in many books, many publications, many lectures, and but his theory is not his theory of gradual incremental differences, gradual evolution from one species to many others, is not supported by the fossil record. And actually, in the fossil record, it supports more like a view of life where there are saltations. The species changes tremendously from one geological stratum to the next. So, however, why Darwinism has persisted? Why is it still successful in certain quarters? Why we still have fervent supporters of Darwinism in academic circles and other circles? It's because we believe that Darwinism contains a half-truth, which is varieties will be successful according to the environment. If they are in an environment where they are fit, if they are adapted to that environment, they will be more prevalent according to the varieties who are less fit. So natural selection explains the comparative prevalence of specific populations with varieties in a given environment. So it explains microevolution, but does not explain macroevolution. For instance, in the Galapagos Island, there is an island called Daphne Major, where in the 1970s you had a drought, and at the end of the drought, the seeds, repartition of seeds differ from the availability of seeds that were small and soft, and some other seeds that were large and hard. After the drought, only mostly the seeds were mostly large and big and hard to crack, and the birds that had smaller beaks, the finches that had smaller beaks, became less prevalent because they could not find enough nutrients. They could not crack the large hard seeds that were available, but the bird here, such as the number one, were more successful, or more predominant in surviving and reproducing. So after the drought, that was found by those findings were published by a biologist couple, Peter and Rosemary Grant, who studied those finches. So the large-billed birds were more prevalent after the drought than before that. So they were more fit to that environment. It's something that that Darwin explains well. Also, if you take the pocket mouse, which could have different coat colors, could have a dark coat color or light colored mouse, and their predators are different animals such as owls and hawks and foxes, coyotes, and snakes, among other animals. But the risk of being put upon depend on the background, particularly in the when they found in the valley in New Mexico. So the mouse, which is more adapted to the dark background, it's better camouflage and better blend with the environment. So it will be less spread upon compared to the light mouse when it's in a dark background. And this is the Pinacate lava flow in New Mexico, where you can find those mice, but they are more adapted. The dark colored mouse is more adapted than that background. However, in the sandy desert that's not far from the the Pinacate lava flow, the light colored mouse is more adapted, and the dark colored mouse will be more preyed upon and will be less prevalent in that environment. So Darwinism explained this partition that you would be more successful at surviving and reproducing if you're more adapted to an environment. But that doesn't explain the origin of the species, whether the finches or the mice at the beginning. It just explains adaptation. So it doesn't explain the origin of those different species. That's our point. So this is and that intervenes our theory. We didn't write our theory by studying the weaknesses of Darwin. We came up with our theory. If you read our book, you will probably we'll write something someday to explain how we came up with our theories. Our theory is the quantum theory of origins that explain, among other phenomena, explain the origin of eukaryotes, explain the origin of biodiversity. What we say is basically it's quantum theory of origin. So the species derive from their quantum phenotypes. But the quantum phenotypes of an eukaryote is a zygote for animal or a seed for a plant. So all the lineages started with their zygote or the seeds in the Precambrian, and those then those zygote and seeds would produce the initial organism, and then those different configurations there will be different configurations from the presentation in the Precambrian to today, and that's how they will evolve from one configuration to the next. If you take, for instance, the human lineage, after the zygotes in the appearance in the Precambrian, let's say we have a configuration A that will later on change to configuration B, later on to the configuration C, later on to Australopithecus afarensis when it came out of the water, later on to Homo habilis, Homo erectus, and finally to Homo sapiens. So, when we look at the fossil record, when we see different fossils of hominins that preceded us, those are not extinct species. Those are species that are previous configuration. Those are previous configurations of the human lineage. And this is our understanding of, so it's so for every lineage that we've seen. So there are no extinctions in that way. Extinctions mostly caused by works of the hominin lineage, such as you can say the woolly mammoth is extinct, but it was caused by an overkill mechanism of previous hominids. And so the quantum theory of origins, when it comes to eukaryotic biodiversity, says that the species of eukaryotes we see today, they are modern configurations of lineages that were began as zygotes or seeds at some point in the Precambrian. So in the Precambrian, there was a zygote and seeds that would change configurations with time, and then so the species or the lineages, they evolve in a parallel fashion. And then those zygote seeds, if you read our book, and also if you look at our previous presentation on this channel, which is the bilateral descent theory of eukaryogenesis or eukaryogenesis, those seeds and zygotes would have been formed by the fusion of viruses, which were RNA viruses, with bacteria. And those RNA viruses, we call them retrophages. Those are retroviruses, and they are an RNA virus. And from that fusion of gamma proteobacteria would form the zygotes and seeds so it's a transformation from prokaryotes to eukaryotes. This is how we think we explain the origin of eukaryotes, from fertilization of bacteria by viruses. So it's not eukaryotes didn't come from archaea, but from bacteria and viruses. And also it's different from the endosymbiotic theory of Lynn Margulis. Again, from the seeds and the zygotes of the Precambrian, we have evolution over time to the species that we have today, and each lineage differently, separately. So what explains biodiversity? First we have the bacteria, and then the viruses. We'll talk about the bacteria. The bacteria is involved. We're different. We have different bacteria for animals and plants. First of all, we have three bacteria that we haven't really yet discovered, thought about the bacteria that led to fungi, but the bacteria that led to all the animals is Margarita namibiensis, which is a bacterium, which is a gamma proteobacterium that was discovered in 1999 off the coast of Namibia by a team from the Max Planck Institute of Microbiology. It was found in the sediments. It's a large bacteria which is visible to the naked eye and that contain a lot of sulfur granules. Namibiensis is derived from bacteria from Namibia, and Tio is from sulfur. Tio margarita, which is mean pearls of Namibia. And bacteria, so all the animals from human to polar bear to snake to dogs, cats, and to foxes or hawks or birds, all of them will have the same. We originate from the same bacterium, which is Margarita namibiensis. Different bacteria for plants, Begiatua bacteria and Superca bacteria. Those were also gamma proteobacteria. And if you go to our book, we'll understand why we place them as the origin of the ovules of plants. So we have first a certain diversity, like at least three bacteria, but the lion's share of biodiversity is caused by the retrophages, by the viruses that were involved in eukaryogenesis. And when it comes to RNA viruses, they do not exist as a single wild type virus. Let's say if you take different RNA virus, let's say the influenza virus, the COVID virus, the HIV virus, or which is a retrovirus. So if you take dengue virus and hepatitis C virus, those viruses, they do not exist as a single genome. So they exist more as populations, as variants that are called quasi-species. It's a quasi-species, multiple variants that are linked to the genome, but they are through mutation. There are different mutations, and they form a population. And then it is that population, that different strains of bacteria or viruses of the quasi-species that will fuse with bacteria to form the different lineages of eukaryotes that we know. Simplified diagram of the viral causative species is very simple. From the wild type of the virus in number 1 here, we have different strains in number 2, caused by mutations at each round of replication. And but the more complex, I said, so here it's a quasi species of COVID virus that was taken from a few samples only. You have the wild type of the virus in the center. After several multiplications, intracellular multiplications, we'll have the quasi species at the periphery, which is different strains of viruses. Just after a few run of replications. So it is understood that it is those different strains of viruses that finally will fuse with the bacterium, with their host, bacterial host, and to form the different eukaryotic lineages. And here this is a quasi-species of HIV viruses, which is retroviruses after obtained from a few samples at clinical sites in Cameroon. The formation of quasi-species is here on the left side. We see a wild type virus that would, after several rounds of replications, that would lead to the large quasi-species we see on the right. Now, again, they come from the initial zygote come from retrophages and gamma proteobacteria. Simple diagram again of viral quasi-species. And it's the quasi-species that will interact, fuse with definitely with bacteria. At number 3, it will cause the different zygotes and seeds that we talked about. So the tree of life that we know of is not a tree of DNA organism, it's a tree of RNA organisms. When we see, let's say, a phylogenetic tree of species, such as this one here, primate phylogeny, it's not that we had an eukaryotic organism that diverge in course, let's say, between bonobo and chimpanzee, or between human and bonobo and chimpanzee. So that tree is a reflection of the RNA quasi-species or viral RNA quasi-species that made us with their fusion of with bacteria, with gamma proteobacteria. So the tree of life is an RNA tree. It's not a DNA tree. After the fusion of the virus with bacteria, and all the lineages evolved in a linear fashion to form from one configuration to the next. The philosophy of Darwinism, which is one of competition and one of struggle, is one of survival of the fittest, while the philosophy of quasi-species is more cooperation. One virus made the quasi-species to its benefit, of replication, a benefit of infection, or causal infection. The different elements of the quasi-species, they complement each other, they cooperate to the goal of infection. Importance of viral quasi-species of RNA viruses, so they are able to resist to vaccines and antiviral drugs because of the different elements that can, different strains that can have different functions and can have different properties. But the elements that mostly show cooperation is during infection, when the quasi-species of viruses, they cooperate for at least one strain to cause the infection. This was illustrated in one study by Vinozzi, which we call the Vinozzi study, a case in point of cooperative interactions among individual members of quasi-species. This was a study that was published in 2006 in the journal Nature by Marco Vignozzi and collaborators. For instance, talk about here the human poliovirus. The human poliovirus normally doesn't infect mice, but we can induce formation of receptors over the neurons of the mouse, so the virus can penetrate the cells in the brain of the viruses to cause infection. So we call that the transgenic mouse model of poliovirus infection. It is made to study the infection in an animal model. So if you take a clone of virus that can cross the blood-brain barrier and infect the neurons of the mouse, if you have let's say 1,000,000 of those clone of viruses that really perform at crossing the blood-brain barrier, and you inject it to the mouse, it will not, if it's a clone, it will not cause infection. It will not be able by itself to cause the blood-brain barrier and cause infection. Now, so we're gonna look at if you make modification to the polymerase of that clone, so it can cause a very diverse quasi-species, very diverse population. And let's say if you increase the mutation rate of the polymerase, or if you further decrease the fidelity of the virus, of the polymerase, or if you tweak the polymerase in a way that it causes a constrained population, it causes less diversity, or less diverse quasi-species. If you inject the clone, which is very performant at penetrating the blood-brain barrier, at crossing the blood-brain barrier and cause infection, with a modification of its polymerase in a way to form a constrained quasi-species, or the quasi-species is very small. Let's say if you increase the fidelity of the virus, or decrease its mutation rate, that clone of virus, even though the clone is very performant, will not be able to cause a blood-brain barrier, because the support quasi-species that it has is very constrained. However, if you make modification to the polymerase in a way to increase the mutation rate, decrease the fidelity of the polymerase, so that clone, that very performant at crossing the blood-brain barrier, which the property the other clone doesn't, the other strain doesn't have, will be supported by that very large diverse population, and will be able to cross the blood-brain barrier and cause disease. So the quasi-species then is on a strength of RNA viruses. So it's a complementing function of those diverse strains that really support that neurovirulent clone to cause the blood barrier and cause infection. I've assimilated this finding to the mission of, I think, Apollo 11 in the 1960s, when you had Neil Armstrong and Buzz Aldrin were able to get into that lunar model and really land on the moon. They were very skilled. Don't get me wrong; they were really skilled astronauts, but they had the backing of the leadership of President Kennedy, the works of different engineers, technicians at NASA. They had the backing of the American people, and they were able to do that. But if you were to multiply the skills of Buzz Aldrin and Neil Armstrong only, that's all you had. Even if you multiply it by 1,000, I don't think they would have been able, with only those skills, they were able to accomplish that mission. So you need a diverse, background of people working in different specialties in different fields to make that happen. That's a cooperation. That's a cooperation of complementing function of strains of population, of diverse population of different scales available, that make the goal happen. Let's say to land on the moon, or to cause cerebral infection or polio infection in the transgenic mouse model, which is different than Darwin, where it is competition and struggle, and it's survival of the fittest. And this view of the history of life, where the organisms, eukaryotic organisms, started first as zygote and seeds, and then that would evolve over several configurations to the configuration that we have today, is supported by the fossil record. So here we're talking about in a region of southern China called the Dushantuo Formation. and we do have those outcrops in black here. Biologists and paleontologists have used the sediment here in those outcrops and submitted them under the microscope and submit them under the microscope, and they were able to find different embryos that embryos found without finding any animals. What they find all those embryos here. We see the 2 cells organism, 4, 8, 16, 32 cells organism that were in the sediments without any animals around. So there was different embryos at different stage of cell division or reductive cell division. So embryo-like fossils and that biota there. And then when they submitted them as well to synchrotron, like a technique called, I think, synchrotron tomography, they were able to find they contain nuclei. So those were not bacteria or the thing. They were really eukaryotic organisms. They were only eukaryotes. They contain nuclei. Here, see, you can see stratigraphy of the terminal Proterozoic region and Cambrian. So at the bottom here, 590 million years ago, you find less than 590 million years ago, we find those different zygotes and seed, different embryos, and later on the Cambrian animals, which were like soft-bodied, Precambrian animals, which was soft-bodied animals leaving just impressions in the sediment floor. And later on you have 509 million years ago, you have the Cambrian animals that were some of them were hard-shelled and living fossils in the fossil record that were found in, let's say, in the Burgess Shale. If you read the book of Stephen Jay Gould, A Wonderful Life, or found also in the Chinese Mao Tianxian shell. So it's an evolution from the left, where you have embryos, to the late Precambrian, where you have those animals on the floor, mostly animal that was sessile, that was fixed to the seafloor, or was really sluggish, and later on, and then those animals didn't really leave fossils, but they leave impressions in the sediments. And later on, you have the arthropods of the Burgess Shale that really fossilized, really hard shell organisms. Some of them hard shell organisms that were found in those different shells. So we have a view, number one here, germinal stage, discovered with microscopy sometime in the Precambrian. Later on, you have the Precambrian animals who are mostly soft-bodied, fossilized impression, and followed by the Cambrian explosion, and then we have many animals with hard parts. So, which give a similar history with the ontogenesis of mammalian offspring. So, similar history than the development, the embryologic development of a mammal or a vertebrate animal, like a human baby, for instance, where a human baby developed after the fusion of the sperm and eggs to form a germinal stage, to form like the many cells organism. Later on you have the embryo, and later on you have the fetus, and the fetus is characterized by the formation of organs. So you have the skeleton that you can see on an x-ray or a scan, but if you were to do a scan for the germinal stage or in the embryonic stage, you wouldn't see anything, which is a similar history than the history of a life at the beginning. For human, you have a zygote to embryo and fetus, and if you were to do a scan or an x-ray, this is kind of a CAT scan of a pregnant woman about 5 months old, 5 months pregnant, and you can see the linear elements of the skeleton of a vertebrate organism in there. It's not recommended to do a scan or x-ray of a pregnant woman, but except in emergency condition, because you don't want to expose the developing embryo or fetus to radiation. You don't want to do that for a child, let alone if it's not necessary, let alone for a developing organism due to the possible adverse effects of radiation. But if you were to do a scan for that same woman, let's say let's say 5 months before, while she was pregnant, let's say a few days or a couple of weeks after conception, you would not see anything. Let's say if you have the embryo, you would not see anything until you have this development of the skeleton. And we think it's the same explanation for the Cambrian explosion, where you have initially the eggs, the zygotes. Usually you have the zygotes or seeds that we can only see with under microscopy, and later on you will have the Precambrian animals that only left impressions on the ocean floor, but didn't have any fossils. And later on in the Cambrian explosion, where it is the same animals of the Precambrians that would make new zygotes, and the new zygotes would really develop the skeleton, the exoskeleton of the animals of the Cambrian. The history of life at the beginning is similar to the biogenetic law of Herbert Spencer, where Herbert Spencer says that ontogeny of an organism, they are very similar. They evolve, let's say, in embryo. The embryonic life from very common to the very from the very general to the very specific. If he takes in his graph diagram where he says the salamander, the fish, the human, and then the first embryonic stage look the same and then diverge in appearance with time, with development. It's the same thing for the animals of at the early stage. Initially, those eggs those zygotes look the same, and later on you have a low diversification appearance with the Precambrian animals that look a bit different, but a lot of the different lineages were still looking the same. And with the Cambrian, started to have diversification in appearance. And later on with the Ordovician, with different other stages of evolution of life, they started looking different. But at the beginning, they did look quite the same, because it's basically it's the same bacterium, Eukaryotes namibiensis, and the same virus with different strains that form them. So it's an evolution of animals from the more common appearance to distinct appearance until we have the configurations that we have today, which is here from the seeds or zygotes in the Precambrian evolution from different configurations until the species or the appearance we have today. But also we have some unicellular organism too. There are some zygotes which did not differentiate into different cells, which remain unicellular, that form, for instance, the unicellular eukaryotes such as amoeba. But what are quasi-species? How are they made? We'll talk about the retrophage, the retroviruses. So they have RNA genome. So when a genome is being copied, so if the genome is being copied, the enzyme that gonna copy it, I'll call it based on the template genome. So if it's gonna use a RNA template, we call it RNA dependent enzyme. So if it's gonna use a DNA template to make another nucleotide, to make another nucleic acid, we call it DNA dependent. So we have RNA dependent RNA polymerase, so a polymerase that we use it RNA template to make another RNA, or we have RNA dependent DNA polymerase, which is a polymerase that's gonna use an RNA template to make DNA. So the RNA dependent DNA polymerase is what we call reverse transcriptase, because they use RNA to make DNA. And then also have DNA, the same process, different polymerases that use DNA as a template. Reverse transcriptase, which is the enzyme that transforms RNA into DNA. So all the RNA enzymes, RNA dependent enzymes, they are characteristically of low fidelity. They make mistakes. They are very error prone, which is different than the DNA polymerase, DNA dependent polymerase. And the DNA dependent polymerase, they have a function they call exonuclease activity, which is involved in proofreading. Let's say if the polymerase insert the wrong nucleotide base, or the exonuclease, call it 3' 5' exonuclease, it intervenes to really remove that mispaired base, and let the polymerase put a new one, which fits to that's in DNA. But RNA dependent polymerase does not have a proofreading function. They don't have the exonuclease activity, so they are low fidelity. So they make mistakes, if you want to call them mistakes. So the DNA viruses, they have, as I said, the proofreading exonuclease activity for proofreading, and the error rate is 1 mistake for each million to 100 million nucleotides copied. So it's very high fidelity. Regular RNA viruses or retroviruses, they don't have the exonuclease. The regular RNA viruses, let's say if you take influenza, COVID virus, or Ebola virus, which are not retroviruses, they make 1 mistake for each 1 to 10,000 nucleotides copied. The HIV reverse transcriptase itself make 1 mistake for each 1,000 to 4,000 nucleotides copied. So they are very low fidelity compared to the DNA viruses. That's why RNA viruses, that's why they make those quasi-species, because they, with each cell division, with each replication, intracellular replication, they come out with modification in their genomes, and then this is that population of viruses that are linked to the wild-type virus that's called quasi-species. As a result of the low fidelity of the RNA-dependent polymerase, organisms' RNA genome replicate with high mutation rates. Thus, after several rounds of replications, they'll form what we call a cloud of mutants, called quasi-species. But how did those organisms, those initial eukaryotes, evolve from one configuration to the eukaryotes to the next, to the eukaryotes today? As we see here, at the beginning again, in the Precambrian, there are seeds or zygotes. They're like form from a fusion of retrophages with bacteria. They will form an initial configuration, and those configurations will change with time to form the species we have today. So if we go back in time, each time we go back, we'll still have the same lineages, but they had different configurations. And so the concept of extension that we find in Darwinism is not that prevalent according to the quantum theory of origins, because those fossils that we look in the past, for human or for birds and the dinosaurs, they didn't go extinct. They changed. So as somebody would say that a bird would fly from the nest of a dinosaur. So so the fossils that we have in the past, they are fossils of our ancestors. They are not fossils of they are bones of the ancestors, which probably should give them proper burial. And for human, let's say we evolved from that initial configuration, the initial phenotype, which was basically like all animals had the sponge phenotype, to another phenotype B, C. At some point would leave the water, would leave the ocean. At some point would be Australopithecus afarensis, at some point Homo habilis, at some other point Homo erectus, and finally Homo sapiens. It's the same for every eukaryotic lineage. So the question is how this evolution was made. It wasn't made according to Darwinian or neo-Darwinian theory from DNA mutation. It was made mostly by what we call reorganization of the genome, and this reorganization of the genome, genetic recombination, is mostly spearheaded by what we call mobile elements, retrotransposons. There are different type of transposons, mobile elements. There are DNA transposons, which are elements in the genome that they can move from one spot to the other, but according to a cut and paste organism. So they would excise themselves from that spot and then place themselves in a different spot in the genome. Those are more prevalent in bacteria, where in eukaryotes you have more retrotransposon, which is those are viral elements. They use a copy and paste mechanism where you would copy those mobile elements. You would copy a DNA sequence, which is basically a non-coding sequence. You would copy into an RNA, and that RNA would move to a different spot in the genome, and then with the help of the reverse transcriptase enzymes would be transformed into DNA. So you have an RNA DNA hybrid, and then the DNA itself would be incorporated in the genome at a different location with the help of an integrase enzyme. So evolution mostly by transposable element and just genetic recombination. Here in red here, and let's say here we have a genome here. In red, this is what we are retrotransposon. It's a mobile element, and also I have to say that the mobile elements were discovered in the 1940s by an American biologist who studied at Cornell University. It was under the name of her name was Barbara McClintock. Discovered the mobile elements while she was studying corns, and she obtained a Nobel Prize for that. So here the retrotransposon here in red would be copied into an RNA, homolog, an RNA sequence, and would move to a different spot into the genome, will be transferred into DNA, will make an RNA DNA hybrid, and with the enzymes reverse transcriptase, and then the DNA would be incorporated into a new position in that genome. So the genome has become much bigger in that sense. So those retrotransposon, they would be involved in the modification of expression of paternal genes, or they will also be involved in creation of new genes. So there are two ways they can act. They can modify the expression of pre-existing genes. When they move to that new location, they can act as a modulator of gene expression. Also, they can be also involved in creation of new genes. Modulation of gene expression also done by retrotransposon, also by the epigenome. The epigenome also, which are as we're not gonna get into that, but modification of the histone on that on which the chromosomes are made in. It's like chromosome that really organize the DNA inside the nucleus. We have also DNA modification as well as DNA methylation, and you have some other mechanism of RNA interference. So there are different modalities of the epigenome, but some of them use the retrotransposon, like mobile element background as well in the act of modulation of gene expression. The creation of new genes can be so the creation of new genes can be done by either gene duplication. Let's say you have a gene A can duplicate into another copy. Let's call that copy B, and followed by neofunctionalization, giving a new function in that copy. So it's just basically duplication followed by neofunctionalization, or it can be by creation of de novo genes. De novo genes are genes that evolved from previously non-coding elements in the genome, particularly from retrotransposon. Let's say if retrotransposon move to a different location, that new DNA sequences, those new DNA sequences, they can form a new gene. It can be like an exon of, it could be like can create a new gene. So those are genes that have no homologs in related species. They call them de novo genes. They used to call them orphan genes, ORF for open reading frames, because they didn't have any homolog in the related species. So the genes creation can be new gene, but we think that the recombination of the modulation of gene expression is the most important mechanism in the evolution of the genome and of the species from one of the lineage from one configuration to the next. And now we have known that human can be actors also in the evolution of the species by using the CRISPR-Cas system. The CRISPR-Cas system was found in bacteria. It's a system that bacteria use to remove foreign genetic elements from the genome, and by cleaving those foreign genetic elements out of the genome, and that same mechanism can be used by scientists to make cuts in the genome of eukaryotes, of any genome of eukaryotes, and then insert new genetic elements. Can be used to treat genetic disease by removing mutations and insert the right bases. And it can be used for the cure of sickle cell or cystic fibrosis. I guess can be used to remove BRCA mutation, possibly. And so the similarity with retrotransposon of the CRISPR-Cas system is that it uses also RNA DNA hybrid molecule to splice the new DNA into the genome. So it's in that the CRISPR-Cas system may be concordant with the way the lineage has evolved before, because it has a similar mechanism of the retrotransposon. And the view of the evolution of lineages, we have shown that how the species, the mechanism by which the species, the lineage evolve from one configuration to the next. But what is the engine? What is the motor behind all this? And even if there's a mechanism, there has to be something, an engine that promotes the evolution from one phenotype to the next. And what we found out that the lineages evolve according to one process, which is basically they evolve from one configuration to the next. The next configuration more able to create entropy for the universe than the previous one. So there's a thermodynamic component in the evolution of species. We call it evolution by thermodynamic guidance, where as the universe is expanding and becoming more disorganized, increasing entropy, this lineages evolve by participating in that increasing entropy of the universe, and call it evolution by thermodynamic guidance, where life process is more complex from energy to invisible heat as it evolves. And for instance, the great transitions of life have clearly shown a pattern of the species and lineages evolving from configurations that were producing less entropy for the universe to configurations creating more entropy. For instance, the transition from prokaryotes to eukaryotes, the transition from a bacterial cell, which was producing ATP or energy to its membrane, and to a eukaryotic cell, which now contains several mitochondria with a greater surface membrane, now creating much more energy, producing much more ATP, several orders of magnitude more than the bacterial cell. The transition from in the Precambrian from animals that were either attached to the ocean floor or that were really sluggish or barely moving on the ocean floor to animals in the late Precambrian that were very active, living in trackways and creating burrows in the ocean floor, and later to the Cambrian animals, where you had Some of them were really ferocious predators like animal eukaryotes, and by the same token, there were to be animals that had to take cover from them. So it transition from less active to very active animals. And later, last but not least, is the transition from ectothermy to endothermy, transition from reptiles to birds and mammals. It's a transition that was made during a time where Earth was even warmer than it is today. So it wasn't made to because those animals were in a cool environment. That stems from that principle of animals evolving from less ordered to more ordered configurations, from animal processing less heat, processing less energy, to processing more energy, to animal producing less heat, less entropy for the universe, so animal producing more entropy for the universe. And we'll talk about the cultural evolution of mankind. We're at a point now we can see evolution to the evolution to the history of our culture. And some would say that that's not biological evolution, but we'll be at a point where we will not be seeing more major configurational changes, major jump in our anatomy and our phenotypes, but we're showing evolution to our culture. Let's say if you take our evolution from hunter-gatherers, our sisters were picking their fruits and their woods for their livelihood, to later development of agriculture. And later on we developed using animal for traction, and at some later point in history where we'll have the industrial revolution with the development of the steam engine, and that we continued on that same pattern with information revolution. And we've been using fossil fuels now for a very long time, where we're creating what we call anthropogenic climate change. And now to the 21st century, where we're acting against gravity, creating rockets like Falcon X rocket or SpaceX to send cargo or human to space. It's a pattern where our history showing that we're processing more energy and creating more entropy for the Earth and for the universe. It's creating, we're participating in climate change as well. And so that pattern led us to consider that although Earth has created, led the emergence of life, but we're transforming our planet to a place that will be uninhabitable, which we will need to live in order for us to survive. We'll need to move to leave the constraints of our atmosphere to go live in space on different planets, and where we'll have the opportunity, the liberty to create, the freedom to create even more entropy for the universe. So this is the pattern we're on, and Earth will not remain suitable for life for a very long time. It's the same situation like if you take an egg, the chick which is developing in the egg at some point had to break through the shell and live, because it has used all the resources of the egg. So this is the pattern we're on, and I think that will continue to that outcome. So here we're saying that with the burrowing animals of the Precambrian, the burrowing animals of the Precambrian, from earlier sluggish animal from near the sponge earlier, the sponge, it was an evolution with the expression of the anterior-posterior axis of those animals. And anterior-posterior axis of animals, the head tail axis is derived from the viruses that made us, because the bacteria themselves, they are round organisms, they are round life forms, while the viruses, they had a head and tail. So so with the expression of that head tail axis, we're able to move from a position A to position B, that able to use and process energy, and we derive that from viruses. I know it's still a question for developmental biology about the origin of the anterior-posterior axis, but it comes from viruses that made us. Also, Hidrotas trackways in the late Eukaryote was showing that use of that anterior-posterior axis, where animals were moving from one place to the other. And we've come to develop an equation for our life history, for the evolution of life, according to which a configuration B of a lineage is producing more entropy for the universe than the previous configuration A, or any previous configuration. This is put in the equation: entropy of E, entropy B of of the configuration B is equal to X entropy of the configuration A, where X is superior to 1. So the entropy production of a configuration B is always superior to that of A. Another expression of this equation is that the differential entropy output between a certain configuration and any of its predecessor, we call delta E. So delta E for B, the differential entropy output for entropy B minus entropy A, it's a positive number. It is superior to 0. So this is an equation for evolution. In that same line, we have developed this diagram for the evolution of lineages, where they evolved we developed it according to the spectrum of visible light, and using the colors of the rainbow, from red to violet, where the lineages evolve from configurations, less ordered configurations to more ordered configurations, from configurations producing less entropy for the universe to configurations producing more entropy for the universe. And we'll conclude by saying that the roots of eukaryotic biodiversity stem from two main factors. Number one, the different gamma proteobacteria involved, and second, the viral quasi-species. The different bacteria involved Margarita namibiensis, to bacteria, and the bacteria that we've talked about. And so we have different bacteria involved in the process of formation of the eukaryotes, and also the different strains of quasi-species, where each strain will be an ancestor for one eukaryotic lineage, a different eukaryotic lineage. And we'll see also that eukaryotic lineages evolve from one configuration to the next, mainly by genetic recombination that are induced by retrotransposons, those mobile elements that move from one position in the genome to a different position using RNA-DNA hybrid. And so the engine and the principal, the vector of evolution of eukaryotes, and for that instance, all life is the need to produce a greater entropy for the universe with time. So species evolve from one configuration to the next, where where the next configuration produces a greater entropy for the universe than the previous one. So this is the vector of evolution and of the history of life. Hence the theory of evolution by thermodynamic guidance. We thank you for your attention, and we will appreciate your comments and your question. And stay tuned for more presentations. And until next time, thank you.